So first, it is my pleasure to introduce Michele Andrasik, Director of Social and Behavioral Sciences and Community Engagement at the HIV Vaccine Trials Network. Michele, take it away.
Great, thank you so much. Can you see my screen?
Yeah, it looks great.
Okay, wonderful. First, I want to thank the organizers for the invitation and the opportunity to present the 702 Behavioral Risk Data Plans. I'll be providing an overview of the 702 demographic and behavioral data, as well as an overview of a recent systematic review that we conducted that is informing our planned analyses. I'll then turn it over to Dr. Crowell, who will provide a detailed overview of our planned analyses. So for 702, the behavioral risk assessment was administered at screening, month 6, month 12, month 24, and the final visit, which was at month 36. All behavioral risk variables were assessed for the past month. These included, and you can see the list here, anal and vaginal sex practices, including number of partners, number of sex acts, condom use, and sex with partners living with HIV. We also collected data on partner characteristics, including age of oldest partner, marital status, whether or not the participant was living with their spouse or main partner, and whether the main partner had other sex partners. We also collected data on sex practices and STI diagnosis, treatment, and symptoms. Each risk assessment also included general risk questions, again, that assessed general condom use, age of oldest partner, parity, age at first sexual intercourse, circumcision status, pregnancy outcomes, HIV and STI test results, and residential circumstances that also confer risk. In addition, we collected comprehensive demographic data. Throughout the trial, interim HIV testing is tracked, and we'll be looking at several of the factors that were assessed in these interim HIV testing, specifically reports of potential exposure, condomless sexual contact with someone of unknown HIV status, or someone living with HIV. And then finally, outside testing and belief data were also collected at month 6 and month 36. And these data include 2 factors that may incur risk, belief of exposure and reports of any new partners. And Trevor will go into this in detail, but we'll also be looking at beliefs of vaccine arm participation, looking at behavioral disinhibition and risk compensation. Before I turn it over to Trevor, who will outline our analyses, the analyses that he'll present are informed by a recent systematic review that we conducted and are in the process of writing up. This systematic review looked at factors predictive of HIV seroconversion in heterosexual adolescent girls and young women living in sub-Saharan Africa. The review yielded 4 articles published between January 1st of 2008 and March 1st of 2018. And as you can see on the right, there were several variables that were predictive of seroconversion across the 4 articles, but there were 2 that really stood out, being age 25 or younger was predictive in all 4 of the studies, and not cohabitating with a regular sex partner was predictive of incident infection in 3 of the 4 studies. So I'll turn it over to Trevor, who will now walk you through our analyses and some additional data that informed our approach.
So before Trevor starts, let me just introduce him if that's all right. So our second presenter is Trevor Crowell, who's the Associate Director of the Department of Epidemiology and Threat Assessment at MHRP. And with that, Trevor, I'm sorry to interrupt, but please take it away.
No, thanks very much, Khalid, and thanks, Michelle. So picking up where Michelle left off, there's a couple more background slides here. One making the point that is intuitive, right? That behaviors are time varying. But what may not be so intuitive is that behaviors may also impact vaccine efficacy. So when Dr. Robb and his team looked at the data from the RV144 study, they saw that the proportion of participants who were classified as quote unquote low risk increased in the first 52 weeks of the study. And you see that represented by this blue line in the figure. Now the distribution between vaccine and placebo arms was similar. So the proportion falling into this low risk group increased similarly among vaccine recipients and placebo recipients. However, vaccine efficacy actually differed depending on which risk groups participants belonged in. So those who maintained low or medium risk throughout the entire study had higher vaccine efficacy, and participants who demonstrated any high risk behavior over the course of the study had lower vaccine efficacy. I also want to bring up the concept of risk compensation. So behaviors that impact HIV vulnerability, when they change over the course of a study, this is a potential threat to the validity of the randomized clinical trial design. And that's the case if they differ in their changes by treatment allocation. This was looked at in the IPREX study of PrEP use. And what they saw there was that behaviors differed based on whether participants perceived that they had been allocated placebo or active PrEP medications. The behaviors changed after enrollment. Similar to RV144, you actually saw a decrease in behaviors typically associated with HIV risk. And the changes were similar between the perceived study arms. So you see similar slopes here in these curves, but you can see that at start and at other time points, there were distinct differences based on whether participants perceived that they had received placebo or vaccine. This was a PrEP study, but I think it's particularly important to think about this in the concept of vaccine clinical trials, because vaccines induce oftentimes a reactogenicity that can be somewhat unblinding to participants, and it may make vaccine trials uniquely vulnerable to risk compensation. So considering these background data, we've proposed 3 analyses here. The first tests the hypothesis that behaviors known to be associated with vulnerability to HIV changed over the course of the study, and in so doing, potentially confounded efficacy evaluation. And our strategy here is first to explore the temporal changes in behavioral risk factors with a focus on those factors that Michele identified from her systematic review and meta-analysis: cohabitation status, number of sexual partners, and others that you see on the screen that have been associated with HIV risk across multiple studies that enrolled similar participant populations. Then we'll use regression modeling to test whether each of these time-updated variables was associated with incident HIV. The next analysis that we'll propose aims to answer a similar question, but taking a very different approach. So here we're hypothesizing that behaviors and potentially clusters of participant characteristics influence vulnerability to HIV and may have also influenced vaccine efficacy. And with this analysis, what we'll do is we'll take an agnostic approach. So rather than coming to the table a priori with factors that we know are associated with HIV risk, we're going to take the entirety of the data that are available from the study, which Michele outlined beautifully, and are quite comprehensive. We'll do quite a substantial amount of data cleaning and preparation for a machine learning analysis to identify key factors and clusters of factors, which may or may not include receipt of the vaccine or placebo that were associated with incident HIV. And we'll use multiple parameters to identify variables that have highest impact in determining HIV incidence, using survival random forest models and minimal depth criteria, variable importance, and also hierarchical clustering to identify not just single variables, but clusters of characteristics that may be important. Once we've identified those most impactful variables, then we can fit everything into a final survival regression tree model. And what this does is it allows you to see almost a decision-making tree of factors that influence HIV risk. And sometimes what you'll see is that certain factors are important within specific subgroups of participants, so higher parts of the tree, and not for others. And it'll be particularly informative to see where vaccine and placebo fit on these trees, and whether they are associated with higher or lower efficacy when clustered with behavioral characteristics of the study population. And then the last analysis, and I think this is also quite interesting, and I was so excited to see that these data were collected in the study, is evaluating the impact of that perceived randomization outcome on behaviors. So we know in this study whether participants believed that they received placebo or active vaccination. We're going to evaluate key behavioral factors as an outcome of regression modeling with that perceived product receipt as one of the predictors. And then we'll also evaluate correlation between perceived and actual study product administered. So the results of these analyses are going to be important for informing the design of future HIV vaccine efficacy trials. Importantly, this includes identifying specific inclusion and exclusion criteria that are going to optimize our ability to detect an efficacy signal in a study population. These will also inform the development of data collection instruments by identifying key behavioral confounders that need to be rigorously measured in future studies. And then lastly, there's the potential to really impact the analytic methods that we propose for upcoming studies. So our analyses will identify whether traditional methods for analyzing data from randomized controlled trials are adequate, or they may miss important efficacy signals because of non-random behavioral changes that influence HIV incidence in study populations. So with that, I just want to, of course, acknowledge everyone who made the HVTN 702 study possible, most importantly, the participants, but also the leadership team and sites. Thank you very much.
Thank you, Trevor. Thank you, Michele. Thank you, Georgia and Dan. So now we're going to open it up for the Q&A session. We have a few minutes. I'm going to ask the audience, please don't be shy. Put your questions in the Q&A box or in the chat, and we'll be happy to answer them. But I'm going to go now to our panelists because we have one question from Peggy Johnson, who I will turn to as the first question for the first question. Go ahead, Peggy.
Thank you, Galit. Can everyone hear me okay?
Perfect.
I'm not sure. Oh, terrific. Wonderful. So first, thanks, everyone. It's nice to see you all. My question is for Georgia and also other panelists. And it really gets at the difference between understanding differences and informing future studies. Because Georgia, you presented a wonderful list of very interesting proposed studies, either underway or planned, that will help understand the differences between RV144 and HVTN 506. 702 rather, sorry. I'm a little, it's a little early here on the
You're designing
West
another
Coast.
trial.
So, yeah. You know, and the J&J trial is well underway. They've got a terrific correlates plan presented by Dan. Thank you, Dan. That was wonderful, and we appreciate the transparency in which J&J is working with this group. So you're all to be congratulated in that regard. But really, what will inform either product development or future correlates analysis? And also with the behavioral work. You know, I like the last slide because it really was forward looking, but you know, it's kind of late to insert any of those into the J&J trials. So if there is another future efficacy trial, I don't think it's going to be for quite some years. So I'm going to turn it over to Georgia first. What really will inform product development versus understand differences? Because I think if you want to get funding, you're going to have to show the relevance to moving forward, not just understanding. If you just want to understand basic research, then, you know, apply for a grant, you know, and it won't be necessarily something that the funders will step up and say, oh, that's really important to do now, because it will inform A or B or C. So if you could just give even an example or two. Thank you. I'll shut
Yeah,
up.
I mean, it's a great question. And of course, the answer is really complex because there's so much we don't understand about protective immunity and the interactions among immune responses. So certainly necessary is for us to really understand the lack of efficacy and the immune responses that either correlated with decreased risk of infection in that study or did not correlate, how those related to prior trials, and better understanding how I guess the differences in people and how they're responding to vaccination. And that is a host of factors that relate to everything we talked about, from host genetics, population, infection exposure, inflammation, all of those things we need to better understand. If you're getting at what do we need for product development, maybe the simplest answer is we need to not ignore studies that don't show efficacy, and we need to better probe what's happening in those trials so we can then further immunogen design. And I think, here, obviously, we're looking at FC effector functions and cellular responses, but I think whatever we learn here will inform all of the BNAB-inducing vaccine regimens, because we're talking about differences in immunogenicity among people, and how do we get everybody up to the same level of response so we can see overall efficacy. And then maybe Peggy, to answer the question about products, that the comparison of once we define the differences and then doing those head-to-head comparisons of products to see which ones are eliciting the best, sort of complex immune responses that we think are correlating with protection status.
So you're saying it could be a down selection tool for future product selection for efficacy studies.
Yes.
If there is one, which there may not be.
Peggy, I'm going to move on to the next question, if that's all right.
Okay, thanks. I could debate Georgia on this the whole session, so.
Well, I'd like to get in on the discussion, but I want to make sure that we get another question out there. So I think the question I have is for Michele and Trevor. I just want to do some rapid fire question here. And that is really, this concept of basically building in behavior to understand efficacy. Can you give us a real world concrete example? How would, when this was applied to RV144, do we see changes in how we interpret the data? And would that potentially have important implications on how we would interpret 702 data that could help really shape the way that, Georgia would be able to evaluate features that could basically respond to Peggy's question in a more directed way?
Yeah, so I could try to take this one, but certainly Dr. Rob or others would know the most about those RV144 data. As a casual observer of the data, I think it is profoundly interesting that you see this difference in efficacy signal based on risk group. And what that potentially suggests to me is that people with lower risk exposure, that's perhaps a lower bar for vaccine-induced immunity to overcome in order to prevent a transmission event. Whereas people in the high-risk group, potentially a higher dose or other type of exposure, that bar is just too high for vaccine-induced immunity as it existed in that study to overcome. So perhaps, go on.
Do you think that the correlates would be different potentially in those two different groups? And would going in to 702 with this type of analysis potentially highlight where these assays could potentially reveal really important nuances that might be different in high risk and low risk populations? And is that the way we should be thinking about these assays that are being proposed, not as pure immunological surveys, but really understanding what is fundamentally different about high risk and low risk exposure, or this idea that you might think you've gotten the vaccine or not?
Yeah, the short answer is yes, absolutely. I think some of these correlates could differ by those groups. I think a key follow-up question is which came first, the chicken or the egg? And I suspect that behaviors might be influencing some of the laboratory results we see, as well as the other way around.
Okay, can I ask one last question, because we have to move on to the next speaker, but I just want to ask Michele a quick question. Would you design a different case control analysis then for 702 and 705, depending on the efficacy signal?
I think that we'd have to really explore the behavioral data a bit more. I don't know if I can really answer that question at this point.
Okay, thanks. It was a hard toss. But okay, I'm going to pass it over to Nicole, and hopefully we can have more discussion about this later.